Hello everyone, welcome to Deepti's biology class. We have already covered microsporogenesis and megasporogenesis, wherein we have seen the male gametophyte is pollen grain and the female gametophyte is embryosac. This is just a recap of what we have already studied in terms of ovule. This is the structure of an anatropous ovule. Just yourself you recall what is the meaning of this chalaza, raphe, polar nuclei. Then this is the exact embryosac, right, where synergids are there, then hilum, funiculus, micropyle, egg is there, right. This is the embryosac. We know how many cells these are made up of. Then antipodal cells, nucellus, integument. If you don't know what all these structures, please watch the previous video wherein we have already covered these things, right. So these are the definition for those who are making the notes. What is funicle, hilum, integuments, micropyle, chalaza, nucellus, and the embryosac, what the embryosac is made up of. For those who watch it for the first time, please understand we have already covered it completely. This is only for the purpose for those who make the notes. So what is the funicle, then hilum, all these parts. When the question is asked, describe the structure of an embryosac, you need to mention all these points, right. The complete, especially embryosac alone, if it is asked, these many points are there, or else if it is asked the structure of anatropous ovule, you have to mention. Why I told you when there is embryosac you need to write about these points, because these all things are connected, right. Embryosac can develop with the help of all these structure. So whenever embryosac even is asked, you have to write about these all points when you try to make the embryosac within the ovule structure or integument. Try to make this diagram only and use this explanation. Now we'll come to the main topic, that is post-pollination event. So pollen grain is ready now. Embryosac which contain egg cell is also ready. Pollination has taken place. We know different agents of pollination, right? How the pollination can take place. Now once the pollination has taken place, what are the events? We call it as post-pollination effect, right? This is what we will see now. Now you remember one thing, we have not yet reached up to fertilization. Post-pollination event is between pollination and fertilization. What is post-pollination event? Between this pollination and fertilization there is a time period. What happens? We call it as post-pollination events. This can include whatever happens after this pollination. Maybe later on there is a stage where the fertilization takes place, right. So let's see the points. Nucleus of the pollen grain divide to produce vegetative and generative cell. These things we have already seen when we have studied the structure of the pollen grain. A small protrusion called germ tube emerges from the pollen. The germ tube secretes enzymes which digest the tissues of the stigma. The germ tube then continued to grow as pollen tube. Hope you remember the structure, right. This was the pollen grain which has these exine and intine and all, right. This cell will divide into two. As a result, what will be there? There will be a vegetative cell and a generative cell. Now after some time there will be a protrusion. What is the meaning of the protrusion? There will be an outgrowth. This outgrowth will grow through this germ pore. What is germ pore? This is the exine, right. The space, this exine is a discontinuous membrane. You can see some spores here, pores are there, right. These pores or holes are termed as germ pore. Through that germ pore there is a protrusion. What is protrusion? Protrusion means an outgrowth will be there that will grow over the stigma, which means, right, if this is the female stigma, this way. So if this is the stigma style, and down there will be somewhere the ovary. So normally what happens, this, if this is the entire structure, stigma is there, right, the pollen will fall here. Pollen will not enter. Instead pollen will grow a tube towards the ovary where the ovule is present, right. This we have already seen little bit and remaining will be clear now. So till now what happened? The pollen grain divides into vegetative cell and generative cell. Now vegetative cell do not participate in the reproduction or gamete formation, generative cell will participate in the formation of the gametes or for the fertilization. This also we know, right? The protrusion called germ tube emerges from the pollen. The germ tube secretes enzymes which digest the tissue of the stigma, and the germ tube then continues to grow as the pollen tube. Now the generative nucleus give rise to two male nuclei by the division. They become surrounded by cytoplasmic masses present in the distinct male gametes. Don't get confused with these points. Just now you make the point as a note, and with the help of the images I will explain what are the meaning of all these points. Now the pollen tube grows through the stigma and passes into the tissue of the style, right? Now entry of pollen tube into the ovule. So what we are talking about, this is the stigma, style and the ovary, right? In the ovary there is ovule. Till now what had happened? There is pollen grain which falls on this stigma region. From there it grows a pollen tube, and the pollen tube has to enter into this ovule. This red colored structure which we have drawn here is ovule, and this blue tube which we have made here is the pollen tube. So the pollen tube has to enter into the ovule. Why? Within the ovule there is embryosac. This embryosac contains egg cell. On the way, what are the changes will happen? We know, right? This consists of two cells. Which are the two cells? One is vegetative and generative. If this cell is the generative one, this will divide into two cells. Which are they? These are the male gametes. How many male gametes are there? Two male gametes are formed in total, right? We'll see that in a minute. We'll see that. Now depending upon the region of entry of pollen tube into ovule, that means we are talking about this area, how this pollen tube is going to enter into the ovule. These are divided into three: porogamy, chalosogamy and mesogamy, and the most common one is porogamy. So we'll see what are these. In porogamy what happens? See, you can see this is the pollen tube, right? This is the pollen tube. Now this pollen tube enters through the micropyle region. We know this is the chalaza and this is the micropylar region. If the pollen tube enters into the embryo sac through the micropyle, we call it as porogamy. If it enters through the chalaza, the pollen tube enters through the chalaza, we call it as chalosogamy. If it enters through the integuments, we know these are the integuments, right? These are the integuments, outer covering of the embryo sac. We call it as mesogamy. So mesogamy means through the integuments it enters. In chalosogamy it is through the chalaza. In porogamy it is through the micropylar region. Mesogamy, even it can be defined as not through any of these extreme point. It can be a semi area. That means from through the side way also you can define. For you to remember, one is through micropylar region, one is through chalaza pollen tube enters, and another one is through the integuments other than chalaza and micropylar region. If you look at this diagram, you can see that the integument is not that developed in chalaza and micropylar region. So which one is common? In most cases, the pollen tube enters the ovule through micropyle. It enters the synergid through the filiform apparatus. What is filiform apparatus? Do you remember the diagram of embryo sac? There are three antipodals at chalazal region. There is a central egg. Then synergids are there. These synergids are having some thickening. What is this thickening we called? This thickening is termed as filiform, right? This thickening is termed as filiform apparatus. Suppose if the pollen tube is coming, this pollen tube enters into one of the synergid by crossing through this filiform apparatus. Then it releases its nuclei. How many nuclei are there? Nuclei in the sense gametes are there. Now how many gametes are there? Two male gametes are there. Right? We'll see what happened to these male gametes. Till now you understand. So which type? If it is micropylar region here, chalazal and here, and this is the pollen tube, which type of entry it is? That is porogamy. Why? Pollen tube is entering through the micropylar. That means there is a pore, there is a gap there, right? If you look here, you can see that pore is there at the micropylar region. This is what we call porogamy. Filiform apparatus guides the entry of pollen tube. This filiform apparatus and everything we have discussed when we studied the structure of this entire embryosac. Now we'll see the next thing what happens after pollination is that pollen-pistil interaction, right? Only the compatible pollen of the same species are able to germinate. The meaning of the sentence is that suppose if this is the stigma of a particular plant. If any other plant's pollen is coming here, will it grow there? If it is some other species, it will not grow. The plant allow growth of the pollen, means we are talking about the pollen tube growth, right? Whenever a pollen falls on the stigma, same species only grow there. That means whenever a pollen falls on the stigma, pollen tube growth is initiated in only the same species. We call it as pollen-pistil interaction. This entire structure is the pistil, right? The female portion of a flower allows the growth of a pollen tube of the pollen of the same species. This is what we call pollen-pistil interaction. If these are of different species, this tube growth will not be there. Germination is related to the action of proteins present over the pollen grains and the stigma that determine the compatibility. So what happens in this situation is that there is a compatibility. Compatibility in another way we can say a match, right? We can say a matching. We say that matching, right? There are some proteins which are present in this style or else in the stigma, right? Now two pollens are coming here. So one pollen, so there are two pollens which are coming on the stigma. Imagine this one is species A and this one is one species B. Now this pollen is having some proteins of this type and another one is having the protein of this type. Now if you look at these two diagrams, you can understand that the protein which are here and the protein which is there inside, both I have drawn with the blue, right? These are of almost same type. They are having some kind of matching. This is what we call compatibility, which means when this pollen falls on the stigma, there is a chance that the pollen tube growth will be there. But if it is coming here, what will happen? Since it is having another protein, this is another species, this pistil will not allow the pollen tube to grow. This is what we call pollen-pistil interaction. How it happens? This is related to germination. That means growth of the pollen tube is related to the action of proteins present over the pollen grains and the stigma that determine the compatibility. Now, plant breeders, we are talking about the artificial methods, right? Obtain hybrid between different species by manipulating the pollination. We are talking about the natural process till now, right? So there, suppose as a farmer, someone or a plant breeder who need a plant of desired character, there he manipulate pollination, which means pollen of the desired plant is collected and artificially or with a human effort it is put into the stigma of the desired flower, so that there is a forced pollination, right? In the female parent with the bisexual flowers, anther are removed from the flower, but before the anther dehisces. Just I will tell you how this is happening, then we'll see the point, right? Now, we are talking about a bisexual flower. In bisexual flower, what will be there? There will be this stigma style ovary, that female portion is there. Apart from the female portion, there will be this male portion. Hope you can understand. These are the male portions, right? Now, if I want to make the petals this way, right? This is a flower which is bisexual flower, right? A bisexual flower is there. In this bisexual flower, both male as well as female portion is there. Now, as a farmer, I want to put the desired pollen here. The desired pollen has to come here. I don't want the same species. This pollen should not fall here. For that, what has to be there? In the earlier stage, from this flower, these anthers are removed. This anther is removed. Now still there is a chance that some unwanted pollen can come here, right? To prevent that, what can be done is that the stigma region can be temporarily covered. If I cover such a way, what will happen? No unwanted pollen will be falling there, right? Now whenever the farmer want to do the pollination, the farmer artificially brings the pollen and this bag is opened. The pollen of desired variety is kept here, then again it is covered. This makes sure or this ensures that only his whatever he wanted, which type of pollination he wanted with the desired plant, only that will happen. So just see the point. In the female parent with the bisexual flowers, the anthers are removed from the flower bud before the anther dehisces. Dehiscence we know, right? If anther dehiscence, what happens? Pollen grain will be there. The pollen will fall on the stigma. So that will be removed using a pair of forceps. Forceps are actually one of the small instrument we can say. In a laboratory we are using it, right? Using that, or simply you can say using the scissors, it can be cut. Now this step is called emasculation. So what is emasculation? Removal of the anther from the bisexual flower so that no pollen grain will fall on the stigma. In the next step, what happened? The stigma of the emasculated flower means these anther is being removed, has to be protested against contamination by unwanted pollen. For that, what will be there? They will be covered with a bag of suitable size to prevent the deposition of unwanted pollen. If you remember, I told you there will be a bag which is protecting this stigma region. Now, the bag is generally made up of butter paper. This process is termed as bagging. Now, what will be there? Whenever this breeder need to put this any pollen of the desired variety, this butter paper bag will be removed and the pollen will be kept here or dusted into the stigma. Again, temporarily it is covered. Why? We don't know whether again there is a chance that few more pollen accidentally comes and that pollen may grow faster. To prevent that, for little more time this will be covered. Now two terms we have studied, which are they? First one is emasculation and the second one is bagging, and you can write one more point, that is tagging. In tagging, what will be there? There will be a tag which contain the entire details. That means on this plant there will be a name board will be there, a small tag will be there. There every details about this plant is written. That means which desired variety we have taken, what is the local name, biological name, all these information, the date, everything will be mentioned. This is what we call tagging. So these things are done actually whenever the plant breeders required it. So this is, if the question is asked how the hybrid varieties can be obtained or how the artificial pollination can be done, you can write about emasculation, bagging and tagging. Emasculation is the removal of this male portion, that is anther. After that there is a bag using butter paper, it is covered. Which part it is covered? The stigma. Then what will be there? Desired pollen is dusted into the stigma. Then if we want, we can keep some information using the tag. That's all for now. In the next section we will be seeing about the fertilization events of these bisexual flower or in the sexual reproduction, right? If you like the video, please share with your friends. Thank you so much for watching.